use the words they like. And we'll argue that they're pluripotent. Others can argue that they're multipotent. It's not so much different. And we'll talk about the use of HAP stem cells to restore function to severed nerves. HAP stem cells can produce nerves in culture. HAP stem cells can produce beating cardiac muscle cells. This was discovered by Dr. Ammel and his group very recently. And it's possible that HAP stem cells may be involved in hair growth, but I'll argue that cardiac muscle cells and nerves are more important than hair. Maybe I shouldn't say this at a hair meeting, but okay. So here's a lady that made the discovery of HAP stem cells. Her name is Ling-Na Lee. She came to work with us about almost 30 years ago and made this discovery of the HAP stem cells by accident. We had these special GFP mice, and we wanted to image stem cells in the brain because they would express GFP, and we couldn't see any stem cells in the brain because the skin was so bright with GFP. And Ling-Na called me into the lab. She said, look, the hair follicle has stem cells, just looks like the brain. So that's how it all started about almost 20 years ago. So here's what the cells look like. They're found around the bulge area. Here they're expressed, this is in telogen, they're expressing GFP. Here's higher magnification. They have a cell body with many of these protuberances here like this. And then Isata came to the lab about 10 years ago, and she did some more high-resolution confocal microscopy of these cells. And you can see here they are in the bulge. Here's higher magnification. You see the cell bodies here and the structures here. Then this Dr. Ammel came to the lab about 2002, yes? You came about 2002, and he took over this project and did a magnificent job. And these are Dr. Ammel's data here, and he would culture these HAP stem cells, and they would form spheres. And the key point is that these cells express nestin. That's how we discovered them. We had nestin-driven GFP mice. And that's how we were so lucky to discover these cells, because they were expressing brilliant GFP in the hair follicles. That means they were expressing nestin, which we expected only to see in the brain. And then Dr. Ammel found that these cells could differentiate and express beta-3 tubulin. That means they are nerves, and they could express keratin. Some cells differentiated to keratinocytes and astrocytes and smooth muscle cells, possibly even melanocytes. This is just the beginning. I think many more cell types, and you'll see in a few minutes a very surprising cell type was found. So one of the early experiments that Dr. Ammel did was he isolated the stem cells, and very easy when you use GFP mice or nestin GFP mice, because the cells are always expressing GFP. You always can check for nestin. That's always your marker. Look for the nestin. And Dr. Ammel inserted the cells into the severed nerves, and the nerve rejoined. And it's very easy to see with a GFP. This is the rejoined nerve here. This is in bright field with GFP, another GFP expressing nerve here. So the nerve rejoined after implantation with the HAP stem cells and can be so easily followed with GFP. So we found that the GFP, these HAP stem cells were forming myelin sheaths here. You can see here clearly, and these probably promoted the rejoining of the nerve. And so then Dr. Ammel stimulated the nerve here, the rejoined nerve, to see if it would cause the gastrocnemius muscle to contract. This was a functional test to see if these HAP stem cells could confer function. And hopefully, oh, the movie's working, yay. Okay, so this is untransplanted. This has a severed nerve, but no HAP stem cells. So you put the electrical stimulation here, and you get the contraction here. And here, this is after transplantation with the HAP stem cells. This is Dr. Amo, he's over here somewhere, and stimulating, and the muscle is contracting. That means the nerve is conducting this signal. So the HAP stem cells are conferring function. And Dr. Amo also repaired the spinal cord. Here's the severed cord here, and HAP stem cells actually growing as spheres implanted into the area of where the cord was severed, and the cord rejoined like this, and you can see the GFP cells in the area of rejoining here. So this is a nice, everything you can see if you use GFP, you always can see. Nothing has to be guessed, you can see. And this is a mouse before treatment. Dr. Amo took this video. I think the video studio was the floor of the lab. And so the hind legs don't work. And then this is a mouse that was transplanted. The hind legs are not perfect, but the mouse can use them and is walking better. So then we started to study human HAP stem cells. These are from the clinic at Kitasato Hospital, Dr. Amo, Dr. Katsuoka isolating them. So these are the nestin stem cells here, and these are the keratinocyte stem cells. The hair follicle has at least 2 types of stem cells, probably more. And here is the area in which these stem cells reside, in the upper part of the follicle, in the bulge area. And you can stain them, because we don't have human GFP humans yet, even though Hulk was in the movies. They stain with an anti-nestin antibody. So they're very similar to the mouse. And they differentiate nicely. These are human, remember now. Okay, so they can make nerve-like cells and GFP, GFAP, glial-like cells, keratinocytes, smooth muscle cells, all coming from the nestin-expressing HAP stem cells from the bulge area of the human hair follicle. And the hair follicle was divided. Dr. Amo divided it into lower, middle, and upper. And the HAP stem cells are most prevalent in the upper part of the hair follicle, as are the keratinocytes. And this is actually a plucked hair follicle. And in our hands, if you pluck the follicle, you don't get the HAP stem cells. Other labs claim they do. We found that if you surgically excise the hair follicle, of course, you can obtain all the stem cells. But if you can get them by plucking, that would be good. Some lab has claimed you can. It may be so. So then the same human HAP stem cells were implanted in mice with a severed sciatic nerve like this, like that. And there it is. The mouse now the nerve is stimulated. The gastrocnemius muscle is contracting very strongly. So the nerve is repaired. So the human stem cells, the human HAP stem cells can repair the nerve too. So that's very encouraging, and we hope we're going to do this in people soon. So then Fang came to the lab. This young lady, you should see her in her military uniform. She's, I don't know, a colonel or something. She works at a military hospital in Shanghai. She spent 2 years with us, and she looked and compare the HAP stem cells in the bulge area and dermal papilla, because they can be also found in the dermal papilla. And we'll see in a couple minutes how they may get there. So Fong was isolating. This is a repaired spinal cord here, and this is from these cells were derived from the bulge area of the follicle. These are mice follicles now, and these were isolated from the. Well, I'm sorry. This is the follicle here. Sorry, follicle bulge area, and here are the stem cells. This is the dermal papilla area. Here are the stem cells. So this is a follicle here. Excuse me. So we could find the HAP stem cells from both regions, bulge and dermal papilla. And here is a spinal cord being repaired here, and these are from HAP stem cells from the dermal papilla. They had previously grown in culture on gel foam, and then the whole culture was implanted and the spinal cord repaired. And these Fong used cells from a red mouse from the bulge area, HAP stem cells from the bulge area, and she could repair the spinal cord with these cells as well. And using a green mouse and red bulge area HAP stem cells, so everything is color coded. Spinal cord here repaired by the HAP stem cells. And I don't have a movie, but this is walking analysis, and these are the mice repaired with either the bulge or dermal papilla HAP stem cells. They walk a lot better than the untransplanted mice. So then Jennifer came to the lab, and she wanted to start growing these stem cells in histoculture, 3D culture. And 3D culture is very good on gel foam. This is a very good substrate to grow anything. The surgeons in the audience will know that you use gel foam as a hemostat, but it also is a great substrate for growing any kind of cell, I think. And so what Jennifer found was that these HAP stem cells, you see here, migrated from the bulge area toward the dermal papilla. That's what this is all about. They're migrating from the bulge area to the papilla area here in culture, in 3D culture. And then Dr. Mee. Is Dr. Mee here today? Yasser? Yeah. Where's Dr. Mee? Are you here? No. Not here? Not here. Okay. Anyway, this is Dr. Mee, and he made great contributions in the lab, culturing the HAP stem cells on gel foam 3D culture. And so here is a follicle here that Dr. Mee started off with, and this is the bulge area, and you can see the HAP stem cells here. They're brilliant here, like that. And he put these in culture, and again, these are the cells here. They go in culture, and the beginning of culture, they start to spread like this. And in culture, they form nerve-like cells and other stem-like cells. They express, for example, P75 here, and they start to migrate. These are neural crest type cells, Schwann cell markers like this here. And day by day, they're growing and migrating. Here they are in culture. This is in culture. This is the shaft. This is the stem cells. And day 1, day 3, day 6. By day 6, you could see them migrating in culture. The processes become elongated. And they're forming beta tubulin expressing cells, P75 expressing cells as they elongate and migrate in 3-dimensional culture. And here, again, showing how they migrate in the culture. By day 8, they have migrated compare with day 0, migrating like this. And they start to form a nerve shaft, a nerve structure here, like this. day 6, day 8, day 12. So this is a new nerve being formed in culture from the HAP stem cells, like this. So nerve growth in culture. And what's driving this nerve growth are the HAP stem cells. And they form a cord-like structure here. You can see this is a nerve cord-like structure formed by the HAP stem cells. So here, this is the nerve formed by the HAP stem cells, and they're interacting with another nerve here. This is the trigeminal nerve taken from a red mouse, interacting with the nerve formed by the HAP stem cells. This is all in culture. You can see the nerves interacting like this, forming like almost like a nervous system formed in culture. And here, this is the HAP stem cell forming nerve is interacting with the sciatic nerve in culture here like this. Here you see, here, this is the HAP stem cell nerve, this is the sciatic nerve, this is higher magnification nerve growing out like this. This is the hair follicle sensory nerve. And, okay, the different markers, tubulin, this is and GFAP, all in culture in these growing nerves. And more here, this is you see all these processes from the nerve that's growing, all growing from the HAP stem cell, expressing beta-3 tubulin here. And here, this beta-3 tubulin stain, this is staining actually for phalloidin, or F-actin, I'm sorry. So it really indicates that this is an axon, all formed from the HAP stem cells, the hair follicle sensory nerve. Here's another, I think, very nice photo of these nerves being formed in culture. This is from all Dr. Mee's work. So this is one of Dr. Amo's recent discoveries. So maybe you can guess what's happening. This is a hair follicle here, and from this follicle, beating cells. So these are cardiac muscle cells. So cells from your hair can form beating heart cells. I think that was quite unexpected and a great discovery by Dr. Amo. And how are we doing on time? Yes? 5 more minutes, okay. So we started to grow, we've made another discovery that completely surprised us. When we implanted the HAP stem cells in the severed spinal cord, the spinal cord started to grow hair. So these are hair shafts coming out of the spinal cord. They were transplanted with the HAP stem cells, just as we did before, to try to repair the spinal cord. And then we took a look and we saw hairs growing out from the HAP stem cells that were transplanted to the spinal cord. So hair growing on the spine, I guess that's a new one. And these are hairs growing in long-term histoculture. I don't know if Mike Philpott is here. Mike is a great pioneer of growing isolated hair follicles. And these are growth curves of isolated follicles growing on gel foam. And what's interesting is as the follicle grows, the HAP stem cells get brighter and brighter. And during the period of maximum growth, the HAP stem cells are the most bright. So perhaps the HAP stem cells are involved in hair growth as well. That would be very nice if that were the case. So I'd like to make a few conclusions, if I may. So the hair follicles contain HAP stem cells. HAP, remember, is hair associated pluripotent stem cells. You could argue about pluripotent, and that's fine. You can argue about everything. That's what science is all about. And the key point, and that's why how these cells were discovered, they express nestin. The other hair follicle stem cells that are much better known discovered by George Cotzarelli. He's here. I don't know if he's in this. George, you here? Not here. But you can meet him. He's at the meeting. He's a great man. And so this was a real surprise that Dr. Lee could find these cells expressing nestin, and we were lucky, just lucky. And that's how it works a lot in science. You get lucky. It's called serendipity. And so we had the nestin-expressing cells, or the nestin-expressing mice, and that's how we, nestin was driving GFP, and that's how we discovered them. And these cells can differentiate, the HAP stem cells can differentiate in neurons, glial cells, and other cell types. And the HAP stem cells can repair nerve, spinal cord. I hope next year we can write here heart. And these cells participate in the whisker nerve elongation in culture, and I'm sure that's what they do in vivo, and fusion with other nerves. And the HAP stem cells can produce beating cardiac muscle cells. This was a great surprise. And when Dr. Amo first showed me this, we were so amazed. And HAP stem cells may be involved in hair growth. I hope so. And maybe some people get mad when I say this, but I'll say anyway. Nestin-expressing hair follicle stem cells are most appropriate for regenerative medicine. This is our hypothesis, and we think they have more advantages than iPS and ES cells because they don't form tumors, and they can be obtained from anybody. Just take out a hair plug. You can come to Dr. Amo anytime or Dr. Saito anytime and get some hair plugs out, and then you can cryopreserve these cells forever. Just put them in some medium that contains DMSO, stick them in the liquid nitrogen tank, and 50 years later, when you need a little regenerative medicine, they're all ready for you. And there won't be any immunological problems at all. So that's our argument. And a lot of people argue against us, and that's just fine. That's what makes science fun. And this is our team here in San Diego. People from all over the world, 4 or 5 Japanese scientists here. English is about the 4th language we speak, 4th or 5th, I don't know. So that's the one issue. If people come to the lab, they don't learn English. And we like our colored mice, and I and mice say thank you for your patience. I didn't expect such a full room today. I'm very pleased. Thank you for coming. And if Dr. Amo permits, I'm happy to take questions.
Thank you so much. Great presentation, Dr. Hoffman. Does anyone have any questions?
He has one. He comes to the microphone. If you have questions, please come to the microphone.
It's amazing cells. Do you know the embryonic origin of these HAP stem cells?
Well, people say neural crest. Is that right?
So it's like Skip's origin?
See, now that's that's what we think. The skip people, they don't like all this. The skip people kind of insist that the cells they look at originate in the dermal papilla. We argue they originate in the bulge and they can migrate to the papilla. I don't know who's right. The skip people are really good, and it could be, and I don't know if there's definitive studies that skips express nestin all the time. So the skip cells may be different, they may not be different. It may depend on how you isolate it and what animal you get them from. We were just lucky. We had the nestin GFP mice, and they were sent to us, go look at the brain stem cells. And Dr. Lee just found them like that. So this is maybe depending on the animal you get them from.
And they express SOX2, another pluripotent stem?
Well, yeah, what about SOX2? A little bit, right?
Express.
They do, okay.
Interesting. The last question. When they transdifferentiate to other cell type, how do you explain that it still remains GFP positive?
Okay.
They should lose the nestin, right?
Yes, and that's a very good question. And Dr. Amo, he solved that problem because after doing his first experiments with the nestin GFP mouse, he said, this is a problem. The nestin gets extinguished. So he started using GFP mice, just driven by beta-actin, because we knew where the cells were. We could see them, we've learned. So by using the beta-actin driven GFP mouse, you keep the GFP. And that's a very good question, and thank you for asking that, or thank you for all your questions.
Do you have any other questions?
So questions?
Microphone, please. When you get to my age, you lose your hearing.
So do the cells leave
Louder, please.
can the cells leave the hair follicle, for example, go to other hair follicles or to form new hair follicles? I mean, in the skin.
Okay. Have these cells been used to form new hair follicles? We haven't done such an experiment, have we? Not yet? Maybe. We didn't do the experiment to see if these HAP stem cells by themselves could form a new follicle. We didn't do yet. We're so involved with nerves and hearts and
This mechanism, neurotheamidin, thus this cells is so easily can differentiate to neural cells. Thus maybe it is little bit different after culture in this condition.
Okay, so you track the cells within the hair follicle. So do you do lineage tracking to see if the cells go to somewhere else, I mean in the skin?
yeah, okay. So if you take these follicles with the GFP expressing HAP stem cells, they migrate from the bulge to the papilla, and they keep migrating out of the follicle. And one thing they're doing as they migrate, they form the sensory nerve. This, I think, is their main in vivo function. So that's what they do. They migrate from the bulge either to the papilla or form the hair follicle sensory nerve. So they're moving. And that's what Dr. Mee saw in culture so clearly, how the cells migrated from the bulge to either the papilla or forming the nerve, the sensory nerve. Yeah, thank you for your questions.
Do you have any other question? Okay. Okay, thank you again, Dr. Hoffman.
Thank you.